And looks like we're live. Wonderful. It's great to be here. It's fantastic. I'm joined by outstanding people, Anna Shapiro, Soya Ganguly, Grace Lindsay, Blake Richards, and Matteo Carandini. And today we will talk about this space relating computation and learning. And I want to start a little bit with the story of how I get exposed to the field of computation. So when I started as a student in Zurich, not even a PhD student, as I was an undergraduate, I think they call it master students these days, Kevin Martin allowed me to watch one of his experiments. And this is one of these super classical experiments where you take a cat, you have the cat there, and a loudspeaker is attached to a needle and that is amplified, and so every time that neuron spikes, you hear a little kick. And so then Kevin Martin would show like bars with different direction, and you'd hear like kicks, kicks, and just seeing that absolutely convinced me that this is the way to understand how a brain works. I think that vision, that this is the way to understand how brains work, are seen with more nuance these days, both by me, and of course that's what happens last, but also generally by the field, both on the experimental side, where if experimentalists look carefully at brain activity, it's not as simple. It's not that I can, for every neuron, I can kind of say, okay, this is the role that neuron takes. It looks like it's experimentally very complicated, but also from a theoretical perspective, this level of specialization, we have a pretty good understanding now that wouldn't even be helpful. And it's pretty clear that this old idea of this strong specialization, and in each part of the brain, there's like one type of cell, or even a small number of them, that do one thing, and we can, as humans, basically figure out what they do and lead to an understanding of the brain, that this old computational neuroscience approach, in a way, is limited. And so, coming from there, now, I think we're in a really interesting time, which is a lot of us are in a way looking for, okay, how should we do neuroscience going forward? And we are really asking us that question. And certainly I'm asking myself that question a lot. And I myself have a lot of uncertainty about how to do that right. And at the same time, we're in this space where now we see two disciplines meet one another. We have the traditional discipline, in a way, based on the Hubel and Wiesel primary visual cortex ways of thinking about brains. But we at the same time have this artificial neural network field that is now progressively building systems that actually work, that gets us excited. And also, along with that came a lot of thinking about how we should think about high-dimensional spaces, not like the singular values of the covariance matrices, of the stimuli. And there's like a lot of different new ways of thinking about brains, and old ways that we rediscover now. And so I think in that context, we're in a really interesting space, because if we do science, it always forces us in a way to look for simplicity. Not like, if the brain is arbitrarily complicated, what are we going to do about it then? So we need to look for places where we can find simplicity. And there's this old idea that it's just what neurons do is simple. But there's a lot of new ways of thinking about it, like where we can say, maybe when we do dimensionality reduction, there's something simple there, or maybe learning might be simple. And in that sense, I think we're in this space. So some other people say, maybe if we look at the covariance matrix between recordings with fMRI, that leads to a simple understanding of how the brain works. So we have these different disciplines that are now looking for simplicity at different places. So now, and as part of that, if you want, we are reminded to think about the conflict between thinking of brains in terms of nature, in terms of how we are born, in terms of our DNA, and nurture about the world in which we start. So I want to, and that leads different of our panelists to different places. And I'd like to start with Blake Richards. So Blake, in a way, you're making the point that a lot of computation approaches will not quite work out, and that the hope is that on the learning side we could make big progress. How do you define this space for you?
Well, so I want to start with a kind of funny counter story to the one you just gave. So when I first decided to shift into neuroscience, I did a master's where we did rotations in different labs so that we, coming from non-neuroscience backgrounds, could learn about neuroscience from different people. And I did a rotation in Wyeth Bear's lab where we were studying visual processing and primary visual cortex. And I was doing computational modeling for Wyeth, but he graciously let me sit in on some of the experiments. And I got exactly that experience you described of, for my first time hearing neurons spike away as each gave the cat different orientation. It was a macaque, I think, different orientations of bars. But one of the things that was really illuminating for me in that experience was also that what would happen is, as we descended with the electrode, we would find a neuron and then we'd run a group of orientations. And Wyeth had this beautiful software that would immediately plot out the neuron's average spiking response to those orientations. And if we got a nice bell curve on the orientation selectivity, we would then proceed to do all our experiments. However, if we didn't, we just kept going with the electrode. And the majority of the neurons that we found, we had to just keep on going with the electrode. And for me, that was really surprising because I had kind of learned this story in my introductory neuroscience class as a master's student about Hubel and Wiesel and how visual cortex was made up of simple and complex cells. And then here, a majority of the cells didn't seem to have clear orientation selectivity. And it for me, it was like, well, something's wrong here about the way that we're approaching this on some level. And I think as I continued to gain more experience in neuroscience and increase my education, that feeling only cemented more and more for me. And what I came to was the feeling that the program that many people seem to envision as the fundamental program of neuroscience, of trying to come up with an explanation of what individual neurons in different circuits were computing, was potentially hopelessly doomed. Now, I should say that particular articulation of an idea that we're going to come up with an account of what every individual neuron is computing in the brain might itself be a bit of a straw man. I think at this point in time, most neuroscientists would actually agree with that statement that I just made. And so it's not that I'm proposing anything radically different from what the field views. But to bring this back to a less straw man argument, I guess what I still think on some level is that when we try to ask that question of what would be the simplest way to understand a neural circuit and to understand it, kind of what it's doing in terms of the algorithms that it's running, the computations that it's performing, how those algorithms are implemented by the circuit, my personal feeling is that the best route into that is to start by asking normative questions about what has this circuit been optimized for. And that can be learning, but it can also be evolution, to be clear. I don't really need to distinguish between the two as far as I'm concerned. And so if you start with this question of like, what has this circuit been optimized for? Then you're kind of doing a top-down approach where you ask the normative question, you try to figure out what the purpose of the circuit is on some level, or what its purpose evolutionarily was. And then you can use that to try to, like a kind of simple account of its function, to try to understand some of the stuff going on internally to it better. And in my mind, that's going to be a way to develop these simple understandings that you articulated, Conrad, that will pay off in the long run. Whereas I think that if we try to go totally bottom up, where we just we trace out circuits, we record activity from cells, and we do this without the top-down normative perspective of optimization, I don't think we'll get to a simple understanding. I think if we do it purely bottom up, we're going to have just this massive, complicated laundry list of biological facts which don't coalesce into a simple understanding. And so that's why the research program I envision is fundamentally this top-down guided one.
So, but if I understand it right, you're not trying to deliver on the original promise. No? So at some level, if it is mostly learning, then if you want, your brain will reflect that Conrad totally arbitrarily has blue hair. He could have green hair, but like it's reflected somewhere. And that means that then there's nothing that guarantees us that the computation resulting from it will be understandable to anyone. No?
So that's part of it as well, I guess. And this is what I think you're driving at here is, I think that the ways in which we can understand a computation or an algorithm going on inside these circuits is precisely with an explication of, say, okay, here is the loss function that was being optimized by this circuit. And so that's why these are the sorts of things we see, because when we optimize neural network models, we see these sorts of things as well. So this is where you can compare the dynamics and the representations in neural network models that have been optimized on these things to real neural circuits to try to develop that understanding. And I guess what you articulated is indeed my perspective to some extent, which is that in contrast, I think if you just try to understand it by looking at the data directly, I'm not sure you can, because I don't think there's necessarily a human interpretable story to tell based on that stuff. I think the human interpretable story is, in fact, this is the loss that was optimized.
Wonderful, very clear. So I was wondering, maybe we should continue with Grace. So Grace wrote a wonderful book, in case anyone in this audience could have missed it, which gives a wonderful overview over the kind of modeling and brain research. So I'm sure she can tell a very nice, clean, high-level story about like what drives her way of doing research here.
Thanks. Yeah, it's good to follow Blake too, because I think I want to be clear about maybe a distinction, this like top down versus bottom up. I think, in computational neuroscience, people already can take a kind of combined top down, bottom up view. It's more normally talked about in like Mars levels way, where you say there's a computational goal to the system and then you try to understand how it's implemented, which is, I think, meaningfully different from saying the system was optimized and caring about the loss function. You could argue that those are different, and then you are shifting the focus of what people should try to understand. The way I think about it is that certainly the use of artificial neural networks, the kind of recent resurgence of them as models in neuroscience, and my own personal research on trying to study them and understand them has made me nervous about the endeavor of neuroscience and the ability to understand complex neural systems using the tools that we currently use. I think it's been easy in neuroscience to always blame how hard it is to collect data on why maybe we don't have the level of understanding that we want. You can't make that argument in artificial neural networks. You can do any recording or any perturbation or any analysis you want to do on them. And so it really puts the onus on people designing analysis methods for what do you actually want to do and what counts as understanding, and do the analyses that you apply. Even if they tell you a nice story in the end, can you then actually use that to do, perturb the network and prove that your story was correct, that kind of thing. And I think it's not coming out great at the moment. It's hard to understand these systems. So I am concerned about the ability to understand computation, but I still believe that there are things that can be said about artificial neural networks and about the brain that we haven't said yet. There are statements about the computations that they perform that are, somewhat simplified versions that aren't just a full description of the data, and that we just need to find the right tools and the right language for those things. And it just might be vastly different than what we're doing now, because as we've talked about, a lot of current analysis methods are still based on the individual neuron and what it responds to, what it's tuned to. Even when you look at population methods, it's still a bit like, how does this population respond to this stimulus? And you're kind of just getting a picture of that. So it could be that we need a radical rethinking of how we understand how to get at computation. But it still is the case that with an artificial neural network, there are a set of weights that will work on a task and a set of dynamics that those weights produce that will make the network able to do the task. And there's a whole much bigger, usually, set of weights that don't. And so there's something different about those things. And I want to believe that we can find that, we can do more than just list the set of weights that work and list the set that don't. I want to believe that we'd be able to come to some lower dimensional statement about what makes a neural network good at something and what makes it not good at it. So I would say that the idea that the goal that we should aim for, or the best we can hope for, is to figure out what the loss function is, that would be kind of the worst case outcome for me. It might be true, but I think there's going to be more that we can do in between there.
So what kind of statements about neural networks, what distinguishes these two regimes, what works and what doesn't work? Now, there's this one set of statements that Blake would probably be very happy, which is, if it does gradient descent on something, it's going to work. If it doesn't, it's not going to work. But you want to have something that basically, yes, and I know I'm oversimplifying this, and apologies for Blake here, but you in a way are looking for something that links the world and the neural network to distinguish what works versus what doesn't work, no?
Well, yeah, if you're going to say, if the ideal is that you can look at a set of weights, or you can look at a set of dynamics in a network and say that network will be able to perform ImageNet and this network won't, then yeah, the question is, if ImageNet has to be baked into that. You do need to understand the statistics of the task that you're, trying to assess the network on and all of that. But the hope would be that, yeah, there's something where, aside from just training a network and just running it to see its performance, there's some way you could look at trained networks and know something about their ability to perform a certain task, because there are big differences between networks that can and can't perform the same task.
Yeah. Great. So why don't we continue from the biological side next? So I would like to hear from Matteo a little bit. So Matteo, I learned a lot because we were at the same place in Zurich where I got my PhD. And he was always doing really cool hardcore physiology. And now he's doing like these amazing big data approaches. Matteo, how are you thinking about this space?
So I was very lucky. I have a big echo. I don't know if you have.
It's only on your side. But we are hearing it. It could be that you have another open window, and otherwise the emergency thing is you just talk and turn off your loudspeaker.
Okay, so I will not listen to myself. So I was lucky to be a PhD student at a time that was magical in the 1990s. I was at New York University with Tony Movshan, and at the same time I was working with David Heeger, who was at Stanford, and Eero Simoncelli was at Penn. And basically these three people figured out a bunch of laws of what individual neurons were doing and groups of neurons were doing in the visual cortex. Neurons were summing linearly from their inputs, which at the time were thought to be in the visual field. They were performing nonlinearity, squaring or half max or something like that. Then they were doing normalization. You have echo too. So I'm going to stop talking and I'll come back to you because I might have another window open.
Wonderful. I agree with the feeling that there was a magical time back then, and we will hear more about the magical times later. Why don't we continue with Anna in this case? So Anna, you've been combining a view on human brains with a view on learning in a truly unique way. What is your view in this space?
Yeah, I mean, so I'm interested in how we learn. Like, that's the question that I'm interested in. So like the answer to that question takes the form of like, what is the learning rule? What is the objective function? So for me, like a satisfying explanation to the kinds of questions I'm interested in kind of takes the form of these like explanations that we're talking about. But I feel like maybe a little bit more optimistic than Grace about like, and maybe then Blake about like what we can understand about these networks and what we do understand about these networks. And maybe that's just because the space that I'm working in, the networks are like more understandable, or like the architectures are more specific, and so that helps us get some like handle on what's happening with representation. So I spent a lot of time thinking about the hippocampus, which has very particular architectures, and if if we defined a particular neural network architecture that matched the hippocampus and a learning rule, an objective function that did a good job of remembering particular episodes, like that would be nice, but that would be really unsatisfying. And I think that we're like already beyond that. So, we understand that there's a certain kind of architecture in the dentate gyrus that has, a connectivity that's very, sparse and different than like just a vanilla deep neural network that leads to this phenomenon of pattern separation. And that means that there's a, orthogonalization of inputs coming in that results in the ability to remember specific things without interference from other related things. And so there's this whole space of thinking about the different parts of the hippocampus and how they would contribute to those kinds of representations that support episodic memory. So understanding the learning rule there is a really important part of the story, but understanding the representations and how they perform the computations that result in something useful like episodic memory is also a really important part of the story. So I feel a little more optimistic. I feel these explanations in terms of learning are super, super important and at the heart of what I care about. But I think we can do more, and we are doing more, at least in this domain, which again might benefit from particular architectures that give us hints about what's happening.
Wonderful. Again, very clear. And Matteo tells us that he's ready for us. Matteo.
Yeah, sorry about that. Senior moment. Okay, so basically at that time, there was this idea that neurons were making these very simple computations and that they would repeat these computations across visual areas. This was all centered on the visual system. But then that program of work got stuck for at least two reasons. One was that it became impossible to describe effectively what neurons beyond V1 were doing in terms of simple operations on images. And the other reason is that it became really hard to explain why on earth, if you show 5 times the same image to a neuron, you get 5 pretty different responses. And so both of these problems are now solved. The first problem has been solved because the artificial neural networks, convolutional neural networks, have shown that if you have enough of these layers, indeed you can get to the point where you recognize a cat from a dog, which at the time we had no idea whether that was true or not. It was just a hope. The other thing that was amazing is that these networks actually use these 3 operations that these 3 guys figured out: linear filtering, max functions, and normalization, which is stunning to me. And then now we also are trying understanding why there's this thing that at the time was considered noise, which is that if you show the same image 5 times, you get 5 different responses. And that has to do with all sorts of learning and interesting things, but also with the fact that neurons in the visual system are not only doing vision, but they're also connected to the rest of the brain. So I think that program of research has been going incredibly well, also thanks to the work of Di Carlo, etc. My personal quest now is to see what else neuroscience can give to AI, for example, and I think there is a little hint of this in some recent work and from Kenneth Harris, with whom I share the lab. We recorded with Carson Stringer and Marius Pacitario from about 10,000 neurons at once, and we showed them 3,000 images, and we asked what was the dimensionality of the representation, and we found that it follows a very, very precise power law. It turns out that with that power law, the representation, the manifold that represents the images, is not a fractal. If it's a fractal, you get the equivalent of adversarial images in convolutional neural networks. But in our brain, we don't have adversarial images. At least we haven't found one yet. And so at least you cannot change one pixel and make me think that you are all at the beach, right? And so possibly that has to do with the fact that the representation follows that power law. So I think the 2 challenges now from an experimental standpoint are understanding what these laws are, such as the power law, etc., and understanding what the rules are, and stop hoping that we can go from stimuli to neural activity or from neural activity to actions directly, because as soon as things get interesting, you can go from neurons to neurons. And I'll just finish by saying that if you took a convolutional neural network now and you tried to understand what the 4th layer was doing in terms of images, good luck. But if you were trying to understand what it does in terms of the 3rd layer, easy peasy. So that's where we are in neuroscience. That's what we're trying to understand now.
Wonderful. And I just want to mention one thing. I think a lot of people here on stage are also interested in what neuroscience can contribute to AI, and I want to keep that out of the discussion today. We probably should have a discussion about it at NeoMatch conference or wherever, about what neuroscience can contribute there. And I think that there's a really nuanced, interesting discussion there. But today, I think I'd mostly want to focus on the other direction. And in that sense, it's great that we have Surya here. One of the things that I really love reading about from Surya is that he really builds ideas on how we can at least understand certain aspects of how neural networks work. And in a way, there's always that hope that the ideas that we can use to understand neural networks might also generalize to brains. Now, in the same way as the things that don't work on microprocessors might be a little dicey at times when we talk about brains, debatable. But in that sense, I think developing techniques to understand neural networks is really interesting if we want to think about how brains work. So yeah, tell us a little about your approaches in that space.
Sure. Yeah. So I guess one thing to say is, like, although I try to be as principled as possible in applying any particular approach to any particular problem, in terms of my selection of approaches, I try to be as unprincipled as possible. Like, the brain is a hard problem. Like, we'll take any method that's useful and we'll try to use it. So I've actually done work where I take Blake's approach and, apply normative models, try to derive from first principles neural circuits and so forth. And then at some point you got to decide, did it succeed or not? And then at some point you have to compare the computation that emerges from optimization in your model to the computation that is seen in the brain. So at some level, you have to compare computation in the brain and the model to assess whether or not that approach is successful or not. And you could potentially compare two computations without understanding either. That's a logical possibility. But to me, that's fun, but it's not satisfying enough. I also do want to understand the computation. And to sharpen that question a little bit, I guess that the crux of the issue is the following. Let's say there's a particular computation you're interested in, say motor control, language, whatever, that unfolds over hundreds of milliseconds, let's say. And the crux of the issue is the dynamical process in your brain that unfolds over hundreds of milliseconds, a fundamentally irreducibly complex dynamical process for which there's no simpler description than that process itself. And I'm loath to believe that that can possibly be the case, because we know that our computations are relatively robust. If they're robust, then their outcome can't depend on the detailed dynamics of every single neuron, which means there's probably a reduced, simpler description. And we'd like to understand that description. Could we get that description in every possible situation? No. Can we get it in certain situations where the answer would be non-trivial enough to surprise us and give us joy? Yes. And the art of science lies in choosing the right problems that are complex enough such that the answer, if we found a simpler description of the dynamics in itself, it would surprise us and give us joy. And so I try to do that in situations where I can.
So can you give maybe an example? So what are the areas? What's the kind of phenomenon for which it can't be complete? It will have to be approximate. So we have the dynamics in the brain. We will have dynamics in our model. How can that link work, or what's the kind of phenomenon that research programs should be able to describe?
Yeah. So the way I like to think about it is the way that it'd be nice to organize research is our goal is not to come up with the most accurate model of the brain, or even a really good facsimile that comes from an optimization problem, right? We have this axis, which is the complexity of our models, and we have this axis, which is the predictive capacity of our models. And of course, a virtual facsimile of the brain would sit out here. It's very, very complex and very, very accurate. And then the question is, as we go down in complexity, we're going to take a hit in predictive capacity. But the question is, how will the curve go? Will it drop like this, which is like the scenario of irreducible complexity, or will it drop gracefully like this? And our goal as scientists is to try to negotiate this Pareto optimal trade-off between complexity and accuracy. And understanding lies in being able to negotiate this trade-off and say, okay, I got a pretty good model, but maybe these aspects of the model are not that important and the final outcome is robust to it. So I can simplify it by coarse graining it somehow, whether it's dimensional ion reduction, whether it's control theory model reduction, whether it's information bottleneck, whether it's renormalization group. Like all these different fields have developed various methods for simplifying complex systems. I think none of them by themselves are up to the task of attacking the brain, but that doesn't mean that we can't develop new techniques for doing that. So now, having said that, it's very important to note that we don't know what numbers to put on the complexity axis, and we don't know what numbers to put on the predictive accuracy axis. Like, what is it that's worth predicting, and what do we consider a complex versus simple model? And that's a matter of research and a matter of empirical. It's going to be worked out empirically and theoretically together over the course of decades, right?
But isn't it also a matter of style?
It's also a matter of style, right? It's a question of what is it that's worth explaining. I remember when I was in a job interview many years ago, I was talking to a synaptic vesicle release person. And in the beginning of the interview, he said, "Surya, we will never understand the brain if we don't understand how vesicles are released." Right. And I politely nodded. But, you know, it's a matter of style, right? I mean, now, having said that, there are many diseases for which very good drug targets attack the vesicle release mechanism, right? So in some sense, he's right for a certain set of questions. But what becomes important, of course, depends on the question you're asking.
So I think this is something that probably most of the panel will agree on, that we're basically trying to find these points on the Pareto surface. But I like for purposes of the discussion to make it concrete. So I'd like to encourage all of you, if you feel that's doable, to kind of sketch how you're gonna, or like which parts of that surface is particularly interesting to you. But why don't we start with Matteo, who
I'm just thinking that we probably all share a hope that there is a decoupling of circuits from cognition, and that decoupling can be summarized by computation. Because if it's not like that, we're kind of screwed. We're not going to understand how. I think we share this. You can tell me if you agree or not. If we can't describe the brain in terms of computation, we then need to understand how 90 billion neurons are connected to each other and in all glorious detail. And it's hard. Now, I think Blake would advocate an even simpler thing, which is not even that you'd have to understand computation, but you just have to understand the learning rule. So, but I think we have this hope. But what if it's not like that? So, for example, to understand how a computer works, it really helps to understand the level of operating systems, programs, and it decouples you completely from the hardware. And right, you don't really need to know anything about the hardware to understand how a computer works at that level. The question we have is whether the brain actually can be explained at the level of computations. And I think some of you would push it further and say, dude, don't worry about computation, just worry about learning. But do we agree on the basics, that we need to understand computation and that might be enough?
So I want to dwell on this, because I think this is exactly like the kind of fundamental disagreement that I think is really important to understand. So in a way, what we just heard is that Matteo would never be satisfied with an understanding of the brain where it's basically, okay, this is what's being optimized. You take it that you can't understand it. The way I understand Blake's point, and correct me if I'm wrong here, is that Blake in a way is fine saying, look, I can understand this is what it optimizes. What it comes up with, I don't understand. So is it fair to say that you have a genuine disagreement in what you mean with understanding here?
So yeah, I mean, I think so, because indeed I am perfectly comfortable with the idea that if we, like, it doesn't bother me in the way it bothers Grace, for example, the idea that we might just only be able to understand the brain through ensembles of loss functions and architecture descriptions related to those losses. Now, to just touch back on the question of this complexity curve, though, the way I'd want to phrase it is, this is ultimately a multidimensional curve. There are many different dimensions on which you can reduce the complexity of your model, and not all of them are going to have equal curvature with respect to that predictive power. And so I actually think that the question that we're debating here, the potential source of disagreement, is which of the dimensions are going to keep us in the nice predictive range, and which of the dimensions, as we reduce the complexity, you're going to see a rapid drop off on predictability. And I suppose that, you know, the thing that I'm articulating is precisely that I suspect that we'll be able to push pretty far on simplifying, you know, the networks that the way that we model networks in terms of many of the biological details of the network and how they operate, many of the details of the computations that specific cells engage in, but that if we push in the wrong direction on like the losses that were being optimized, we'll just boom, lose predictive power immediately. And so that's why I feel like, to some extent, I'm comfortable with models that maybe, and also attempts at understanding neural circuits that maybe don't always focus on a lot of these other things, and instead focus on the question of what's being optimized, precisely because I feel like that's the dimension where we're going to get the most bang for the buck in terms of this complexity curve trade-off.
So am I right with assuming that the sympathies of the remaining three of you, Grace, Anna, and Surya, lie with, at some level, you want to deliver on Matthäus's promise, no?
I mean, in terms of, you know, where on the complexity curve you fall, I think it is a matter of what question you're asking. I mean, I think ideally we'd be able to kind of zoom in and find an answer that falls on any point for any system, you know, that people are studying. Because on some level, at least for me, you know, if you want to offer something that explains the whole brain, I think the universal approximation theorem that says that infinitely wide neural networks can implement any function, like that's a pretty satisfying answer to me. That grounds me in believing that brains can do intelligent things because they can implement any function. And so that's just a broad statement that can explain the whole brain. But then we go into, okay, what is the actual neural network that, you know, is doing that, and what is the function it's implementing, and how does it learn the weights that it needs to have? So it's really, I think, people can ask questions at any level and they're going to get different answers. Sometimes where the problem comes is people have a misalignment between the level that they're asking a question and the kind of level at which they're applying an analysis that could produce an answer. If there's a misalignment there, I think that's a problem, but you should ideally be able to kind of get any kind of model you want at any point on that curve, if we've kind of fully done neuroscience.
So what about the rest? Wait, Anna, what's your view?
I am very sympathetic to Blake's to approach that, like, we will make a lot of progress by starting top down and thinking about the optimization problem and the learning rule, and so I'm very sympathetic to that. And then I don't think Blake disagrees. Like, I just think we shouldn't just give up once we come up with the model that does a good job of predicting, you know, if we have that model, right? Like, we can then understand the model, or we can endeavor to understand that model. But I basically am
well, so in that case, could you or Blake give an example of success of that research program?
I mean, I think that well, I mean, I'm not a vision person, so maybe I shouldn't say this, but I think the correspondence with these convnets and IT cortex is amazing and inspiring, and the kinds of representations that you see in these networks, they to some level of approximation makes sense, right? You can look at them and say, I understand that there's a certain similarity structure here that that's useful for the task that they're performing, and so that's yeah, I think that's for me, that's a big success. Yeah.
I would want to add one other success to this. So, you know, I think it can be very hard to interpret a lot of neural data related to action selection and the response to rewards. But once you start viewing it through the lens of reinforcement learning and optimizing for maximizing rewards with a bootstrapped estimate of value, all of a sudden a bunch of neural data makes sense. And that's an example of where a top-down perspective really helped to make previous data that was very hard to interpret interpretable, courtesy of a specific in this case, not a loss, but a specific thing that you're trying to maximize and optimize on.
So I briefly want to ask Alana Jones's question, because it's been very popular in that list, and she would probably kill me if I don't ask her question. So she asked, in a way, she'd like you to be more precise about what you mean with understand. And I think that, like, Surya's understanding is just navigating this complexity versus explained variance trade-off. That's not all of it. They understand means something much deeper, I think, for us. So why don't you all chime in kind of what it means for you to understand here?
Yeah, I mean, Richard Feynman had a nice kind of explanation of, like, you know, you understand a theory if you can say something about the solutions to the equations of that theory without actually solving the equations, right? So, like, case in point, like fluid mechanics, right? If the only way you could predict fluid flows is by numerically integrating the Navier-Stokes equations, then you'd be loath to say you have some intuitive understanding of fluid flow, right? But we can actually predict at a coarse grain level approximately what will happen in many fluid flows, right? Similar to that of a complex neural model, like let's say we have a really good model of the visual circuit, right? But we can't explain how it will respond in certain situations qualitatively. Then I'd be loath to say that we sort of understand it. Actually, we faced this problem with the retina, right? We came up with, like, to our knowledge, the world's best model of the retina to natural movies. Okay, we trained it on like 20 years of we tested it. We trained it in natural stimuli. We tested it on 20 years of experiments, and we got all those experiments right. One of them was the emitted stimulus response. You show a full field flash, the retina will fire. And then suddenly you emit a flash, the retina will fire even more, right? So it's as if the retina learned the predictive model of the world, and then it signaled a violation of that model, of that prediction, right? Why is our big neural network giving us that response? It's a qualitative question that demands a qualitative answer. And we needed to do further work to extract why that was happening. It involved model reduction, a judicious algorithmic approach, reducing the complexity of the model without throwing out its predictive capacity for one particular stimulus, right? So things like that, I think are, and then we felt satisfied, right? And I'll throw out a thorn in it. Understanding is fundamentally a subjective human quality. It doesn't exist outside of the mind of the person who claims the understanding. So in some sense, we as scientists maybe know better than artists. Understanding is like beauty. Artists are in search of it. They kind of know it when they see it, though they have some disagreements about it. And that's okay, right? We're kind of entertaining each other with our own sort of understanding, and we hope that other people will appreciate our understanding. And so we have to remember that the pursuit of science is in some sense subjective. So I'll just throw that bomb out there and leave it at that.
So it's a bit about unexpected predictions. Arguably, much of art has that too, no?
Yeah, it's a bit about that. But I think the heuristic is the following. It's like an Occam's razor heuristic. If we can explain the most amount of experimental data with the least amount of theoretical input, we tend to love such theories, right? And so that's what we'd like. You know, that's kind of what we try to strive for.
I totally agree, and I think, in fact, that people work well together when they agree on what constitutes an answer to a question, right? And so you and the vesicle scientist might not be good companions because you disagree on what constitutes an answer, right? And or you might be great companions. I don't know, but what I'm fascinated by is whether there is a continuum of levels at which one could say I'm satisfied with the answer, or whether there are discrete levels at which things decouple. So from my very limited understanding of physics, of solid matter, you can have equations that are fantastic when you have a couple of particles or 3 particles, but as soon as you have like 5 particles, these equations stop working. And then things start working again at a totally different level of not even billions, but like lots of 10 to the somethings of particles. And then you have fantastic equations again. And so in between those 2 levels, you better not hang out in between those 2 levels, right? And so I'm wondering whether in neuroscience we're in a similar situation. So the vesicle person is going to have that level, and maybe there are valid answers at that level. And then maybe there's a bunch of levels where you really don't want to hang out. And then there's the circuits person, and then there's Surya, and then there's Blake. But you see what I mean? And I'm curious whether there's decoupling. And if there isn't decoupling, I think we're in deep trouble. And so similarly, you know, if you want to understand the economy, you don't need to think about the atoms of the individual people, right? But there's various levels in there that decouple things and that help. Now, I'm not sure we understand the economy that well. Hopefully we'll understand the brain better. Anyway, I better shut up. I spoke enough.
So I was wondering, Grace, you wrote a wonderful book, in a way summarizing our field for the world. What would you say is like the overall summary of what people mean with understanding in the field? Now, you must have seen certain common trends.
Yeah, well, usually, I mean, the book is centered on mathematical modeling, which always involves letting go of some details. So the trend is usually that you acknowledge that you don't have to understand certain components, or, what you're trying to understand it's going to determine which components matter or not. So I think this notion that Surya put forth of, this ability to predict without having to run a simulation, to actually in your mind predict, is related to sometimes why people build mathematical models or like mathematical models, because they're these reduced abstract things. Yet ironically, they're also the things that let us run simulations so that we don't understand. So it can definitely go both ways. The models themselves don't make it so that you get an intuitive understanding. And I think that's why we started this talking about artificial neural networks, because you could train them, you've built them entirely, but you don't know how they work. At least, that's the common refrain. So yeah, I think the idea that understanding is different to different people and different in different circumstances, and someone who studied a single cell type in the fly for 20 years can say that they have an understanding that I won't be able to have because I'll need 20 years to get to it, or whatever it is. They still have that understanding. They could probably have intuitions and predictions about, what that cell type will do under different circumstances. And so we just have to accept at some level that understanding is different for different people, and different for different questions. I would just go back to the notion of, the problem occurs when within a person, within a research area, there isn't clarity on what the goal is. I think that's what the issue is in a lot of cases.
Yeah, absolutely. So we only have a few more minutes, but I want to highlight another dimension where I think there's significant disagreement within the panel, and where we can benefit from some clarity. Like Blake said it very clearly, like he has a top-down view. So in a way, a top-down view is, I think, the way, and again, I'm putting words into Blake, but in a way, like, I think the way Blake is thinking about it, what's the problem that's to be solved? Say, be as efficient as possible at solving a given pattern recognition problem or something, at detecting objects or something like that. And then he starts with that and basically uses that to go towards real phenomena. And so at some level, systems that work is what everything is built on, because you cannot build top-down models unless you have really built systems that work. And then there's the bottom-up. Let's look at, as Matteo so nicely put it, like there's these rules if we look at individual neurons. So where does everyone stand on this axis? And how useful are those two trends for you? And I'd like to not start with Blake or Matteo, because I think for them, we kind of understand at least where their personal history ran. Maybe we should start with Surya in that case.
Sorry, I missed the key thing about what the two axes were. I was looking at the chat.
So it's just now about the use of top-down thinking. Basically, what's the problem and how can solutions look like, versus use of bottom-up thinking. Here's what individual neurons do. What does that mean about bigger principles?
Yeah.
No, there's
I'm completely unprincipled about what approach I take. I mean, I use the top down, I use the bottom up. We got to make them meet. Like, everybody should work together, hang out together, like exchange ideas. I think, I think it's extremely important to confront the top down approaches through experimental tests with bottom up details. and bottom-up alone will never get us there. Like we have to like think about the reason for the existence of a brain circuit in order to understand it, right? Like yeah, so it's yeah, I mean, there's these famous quotes, right? Nothing in biology makes sense except in the light of evolution. The analog of that in neuroscience is nothing in neuroscience makes sense except in the light of behavior. Behavior itself might not even make sense within the context of the evolutionary arms race that evolved that behavior. So all of these levels are interlinked, and I think I noticed in the chat there was this crazy comment made by some PI that bridging levels is not important. I think bridging levels is the lifeblood of science, right? Like physics have this in stat mech to thermodynamics, like engineers have this in going from transistors to computation. They do it by trying to decouple levels as much as possible and building in abstractions. Evolution has done it. It is built in robust systems with chemical modules, cells, circuits, brain regions, and so yeah, I think by bridging levels you can go both ways, and you have to. And woe to the scientist who picks only one way to do it.
So Anna, what about you? How much does your thinking about brains, how much is it influenced by thinking about the problem to be solved versus the data that we get out of neuroscience?
Yeah, I said this earlier, like I tend to be more top down. I mean, maybe that's partly because I study humans and our methods are a little coarser, and so it's like easier to be top down in a way. But I mean, like we need data. Like we need data early in the process, not at the end of the process, right? If you wait until you have your model and then it's all built and it's all optimized, and then you say, okay, like now I'm going to go and check and see what's happening in the brain. Like that won't work, right? We need a lot of information about the architecture, right? We probably need a lot of information about the way that like possibilities for learning rules before we even like start to make the model. So like it has to be a back and forth, right? Like it has to be a back and forth, and so there are things that we know about the brain from like dumb, boring, exploratory, like let's just look and see what there is, and that informs our model building in really important ways, and it goes back and forth. But the kind of research that I like personally am engaged in and find like most exciting is the kind of research that I guess. Well, yeah, I mean, I'm glad there's all kinds of people, right? Like Surya is saying, I'm glad there's people who don't think this way, but the way that I think is a little bit more on the top down side. Let's define the problem and the learning rule, and let's see what kinds of representations we get. Now let's go check in the brain and see if we see those representations. And okay, and now let's try to understand like why do we see those representations? Why do they solve the task? And so I tend to be on the more top-down side, but I think we need data all the way, and I'm sure Blake would agree with that.
Maybe as a last person, Grace, where do you find how much do you find yourself using top-down versus bottom-up thinking?
I think I'm very squarely in the middle because, like Surya, I think that the fun of science, and particularly neuroscience, is connecting the levels. Like that's how I got into neuroscience. Like you can connect cells and they produce behavior, and like that's crazy. So I like to be right in the middle, but I'm perfectly happy to have people who are very isolated in their own level, because then I get to pull from their data to do the connecting myself. So I like reading, a purely behavioral psychophysics study and a purely, anatomy of the visual cortex study, and then building a model that tries to link them. So I think, yeah, we need people to do all different kinds of things, but to me, the fun answer is how do these things connect?
Now, unfortunately, we will need to stop roughly here. We could easily have spent the whole time taking a small part of the overall puzzle that we've been talking about. I want to take the time to thank all panelists. I love the level of clarity and also the fact that you all, in a way, totally agree with one another, which is good in a way. And I hope that was useful for everyone who saw it. And so let's thank all the panelists again. It's the first time you get to hear like not very loud clapping here. And thanks for participating, and thanks for the audience for the great questions. Thanks so much, and bye-bye.
Thanks for having us.
right, hello everyone. Hi, welcome to the 8th short talk session. I'm going to be, I'm really pleased to have 3 talks that we'll be presenting today. I'm going to start off with Harak Asmirian, and he is at the University of California, Berkeley, and he's going to be giving us a talk about motor learning. So go right ahead, Harak.
Cool. Share my screen. Can everybody see my screen?
Mm-hmm.
Cool. Can't really seem to see you guys though.
Oh, that's okay.
Cool. And you can hear me all right?
Yeah, if you could click on that hide button too, that would be useful.
Most definitely.
Perfect. Thanks.
Cool. So my name's Harach, and today I'm going to be talking about motor learning and how we can identify some predictors of motor learning using a data-driven approach. So what's motor learning? Motor learning is anytime you use your motor abilities to learn something new and to adapt. So for instance, if you want to reach from some starting position to some target, you might fling your hand forward and notice that you've made some kind of error. Subsequently, you adjust to this error and so on and so forth until you complete your task and you adapt your motor system. And in the lab, this is typically studied using the apparatus you see on the left. So there's a desk setup where users are seated with a stylus in their hand and a digital display that hovers over the desk. And they can see where their stylus goes as identified by the white cursor on the screen. And in recent months and years, the Cognition and Action Lab at Berkeley has innovated methods, and now users can do this experiment online from the comfort of their home using an optical mouse or a trackpad. And this opens the door to many more users because this is so accessible, and also a greater diversity in people who take this kind of experiment. So much broader reach for this method. So typically, one example of motor learning task is the one shown on the right here. So initially shaded out in your hand is that you're reaching for some target and your cursor is offset by some amount. There's some degree of rotation imposed on it, but eventually you adapt, you reach in this case to the right to get the cursor to the target. Well, this kind of adaptation is typically studied along some axis. So how does this adaptation vary with respect to, let's say, cerebellar ataxia, which we see on the left? So on the top 2 slides, you see the control subjects. It's a very standard adaptation curve once this rotation is imposed on their cursor versus their reach. But in cerebellar patients, we don't see that kind of adaptation and they plateau. And so this is one axis along which you can study adaptation. And another is task features instead of neurologic features. So Jonathan Say in the lab has worked extensively on this, working on what kinds of feedback on your screen influence how you adapt. So there's this whole space of features. And a big question is, how can we identify the important features for motor learning? And how can we let the data speak for itself instead of coming up with hypotheses and addressing them more individually? Well, first you need a task. So we developed a motor adaptation task. On the left, you're seeing average performance on these dots. And we recruited about 1,500 people. So this is a very, very substantial sample size in motor learning research. And on your screen, you'd see what's to the right here. So you move your cursor to the center and you reach for some target and you adapt. You get used to doing this task until you've gained some proficiency. And then we impose a rotation, just like I showed you earlier. Now, when you reach for the target, your cursor goes in a different direction and you have to figure out how to adapt. And that looks like what you see on the screen here. Your initial reaches are very far off until you figure out what's going on and put the pieces together. And on the left, you see the standard adaptation curve that correlates with this. And in the last phase, we instruct users to just reach back towards the target, but we don't give them feedback. So they can't adjust to errors. And we see residual effects from this adaptation rotation phase. So now that we have a motor adaptation experiment, we sought to see, is this a consistent metric of adaptation? So we compared the even and odd reaches of users over these different phases to see if they hold. So if your even and odd reaches on this task are not correlated whatsoever, then this can't hope to be a good metric of motor adaptation in the online world. But what we end up seeing is the opposite of that. Even and odd reaches along these different phases are very highly correlated, and this provides good within-user consistency. So this is a good metric. Also, we calculated Cronbach's alpha, which is another psychomotor consistency metric, and we got 0.92, which is fairly consistent. Also, Jonathan Say in the lab has worked extensively on this as well, showing consistency of online methods versus in-lab methods. So now that we have this performance in reach, we look at the distribution. Along this early bracket, there's a fair distribution in performance between people. And same goes for late and after effect phases. So now we have a metric and we have differences with which to explore the feature space. And now we need features. So we collected roughly 20 demographic feature questions at the end of the experiment. And this revealed that there's a roughly equal split between optical and trackpad users. We recruited over 100 left-handed people, which is really novel in motor learning space. It's really exciting. And additionally, a greater diversity in racial groups of people who took this experiment, though still predominantly white and Asian, and an equal split between males and females. So in addition to these demographic features, we collected some task info in this baseline phase, like what were reaction times and movement times in the experiment. And we have all of these features, and we sought to predict one with the other. Using all of these demographic and task features we've accumulated in this space, how can we best predict motor adaptation outcomes? So we carried this out using a multiple regression method, namely Lasso Regression. And this method adjusts the weights within the model to best predict your outcome, and it shrinks some weights to zero. So it eliminates some variables from the feature space implicit in the method. So we use this method and cross-validate it to get the right hyperparameters so we have a good generalizable model. And we sought to predict early, late, and after effect phases of adaptation. So the result of these models is an explanation of variance between 2.4 and 11.8%. And what you see on the bottom left is 1,000 models generated on permutated data. So if I randomize demographic features and create models using this randomized set, how well can I predict actual outcomes? And it turns out, obviously it's very poor, and our predictions do much, much better than this baseline. Additionally, other studies in the same space of predicting a motor outcome using background information and task information capture variances in the same range. In this study in school-age children and motor adaptation, it's between 9 and 13. Though some other methods do get higher degrees of variance, I think it's important to note that these other metrics have vast batteries of motor ability, whereas we're studying the swipe of a finger or the movement of a mouse using background and task information. So I think given the task we're using, this is really substantial. So given that we have a model that can make these kinds of predictions, we can analyze the weights and see what does the best model use to figure out people's adaptation performance. And this is kind of a lot to take in. There's a lot of plots and a lot of different weights in early, late, and after effect phases, but I'm just going to zoom in on some of the ones I thought were important. So one are noted gender differences. So on the left, I've highlighted the features that fall under the group category of gender, how people reported it, and how that correlates with people's actual performance. And on the right, you see reach behavior separated by that metric. Though this is far from surprising given the history of motor learning research in this field, gender disparities have been found, and there's been a lot of work on gender stereotyping and training in the effects of this. And I think this provides credence to the model. This interfaces with the broader motor learning literature in a consistent way, at least in this way. But additionally, in addition to confirming some canonical findings, it offers areas for investigation we otherwise wouldn't have really looked into. So one of the most strongly positively weighted features was how people rated the experiment afterwards. So a 5 out of 5 rating would be satisfaction, great satisfaction with the experiment. 1 out of 5 would be poor. And we see a very close correlation with how well people did and how much they enjoyed the experience. And that wasn't really something we were thinking about testing beforehand or we thought was relevant. And the same goes for movement time. So how fast do you move your target from the starting position to the target? It turns out that the people who reacted faster had more accurate performance, pretty dramatically, if they were above the median, and vice versa for longer movement times. So these provide some new areas for investigation where you wouldn't really anticipate differences. And additionally, features that are removed are also really important. So one would expect that there would be some difference between mouse and trackpad performance because it seems to be a different movement. One is with a finger and one is more gross motor activity. But the fact that there isn't much difference along this axis says that this is a pretty consistent method. This is a reliable way to study this motor adaptation. Additionally, major didn't really seem to interface with differences in adaptation, which was also interesting. And very quickly, in this after effect block, as you see on the right with the black bracket over it, the greatest differences were dramatically found on the direction of the target. And this was a very unexpected finding, and we weren't really thinking about this direction at all, but this shined a light on this influence. So roughly, whether or not your target fell in diagonal directions or cardinal ones, which is north, south, east, and west, made a huge difference in this residual implicit component of motor adaptation, thus shining a light on something important we otherwise wouldn't have really come across. So just to wrap up, this method of regression and selecting features within it shines a light in the dark space of all of the features. And sure, we can't capture all of the features out there, but this is a big step from an individual hypothesis test. And additionally, it's data-driven. The data speaks for itself. You don't need to have all of these priors and one very explicit question coming into it. You let the model paint the picture of what seems to be important. Also, it reveals novel features you would have never anticipated being important that can eventually paint directions for future research. And yeah, I think this is really exciting work. And I want to thank Jonathan and Rich from the Cognition and Action Lab, as they've helped my thinking along this line. And also Laura, Jeremy, and Ken from testmybrain.org, which is where this experiment was initially offered. Also, if anybody's interested, you could take this experiment. It's online right now. If you follow this link, multiclamp c2 web.app. And I also want to thank Neuromatch because I think this is such a great opportunity for people to see the work that's out there and get constructive feedback. And yeah, I just want to say thanks a lot for the opportunity.
Wow, that is really fascinating. I can't believe that you can get such great data out of an online forum like this. That's great.
Seems like I'm getting a feedback loop here.
Uh-oh.
Whoa. Do I not share? This is strange. Oh, there we go. I'm back. Sorry about that. Oh, I think you're muted.
So that was great. That was really fascinating. One of the questions that I immediately had in thinking about the fact that you can do trackpad versus mouse is whether there are residual after effects if you train them up on trackpad and then have them switch to mouse.
Yeah, that would be.
Have you tried that one yet?
No. So in this experiment, people just log on and report what they used. So we don't really designate what people should or shouldn't use. On the website, it says recommended trackpad because that was our initial line of inquiry, but it ended up being 50-50.
Right.
But I do remember reading a paper I forget from when, but there was a disparity between different ages and using a mouse versus a trackpad. And I didn't see these effects clearly laid out in my data, but also great line of inquiry. Like, because they are different motions, this calls into question, like, is it really the same underlying system that we're calling upon? And I wonder if switching between methods would illuminate this.
Well, yeah, absolutely. Do you mind also saying, talking about what software stack you used for this? Was this,
I use RStudio and a fair set of the machine learning packages like GlimNet and all of those.
Oh, no, sorry. I meant for actually running the experiments and collecting the data online. Was that JavaScript?
It's called OnPoint. I believe it's coded in Java, and it was developed by some graduate students in my lab.
Okay, great. Yeah. Is that something that's available for other people to use?
Yeah, open access.
Great, fantastic.
Yeah, great tool. Really, sample size is the big contributing factor here. In motor learning research, how are you going to recruit 1,500 people to come into your lab and move a stylus? So I think this paints the way for larger big data approaches to these problems.
Absolutely. We got just a little bit of time for questions since we started late. John Patterson asks if you looked at the history of gaming.
Yeah, one of the questions we asked was how many hours a day do you play video games? And we mapped that as a continuous feature. That was one of the weight features in the model, positively predicting. So more hours of video game playing is a positive predictor of adaptation. Although I think it's important to know, I didn't have time to really dive into this, but a limitation of multiple regression is that if you have two features that explain the variation identically, no model is going to tell you which one is the real proximate cause of it. And I think this is underlying deeper problems and what we mean when we say, oh, there's a close correlation between these features. So for example, I had this problem with age and education. They were very, very closely correlated as a correlation matrix revealed. And this poses a problem. So for example, education was included in my model, but age was dropped. And does that really make sense? It's a shortcoming of multiple regression. So if you use other tools like a correlation matrix to highlight these questions, which I'll do in the discussion, you get around this problem.
All right. Well, in the interest of time, I think we're going to move on. Sorry, Franz, if you want to ask that question, you could maybe bring it up in the Discord channel associated with this talk. By the way, the Discord server is not going away at the end of the conference, so please feel free to continue to have conversations there as we move on. So Harach, thank you very much. That was a great. I just love the idea that you can collect data in this format and then it has such validity. That was great.
Yeah, thanks so much.
Okay, so next up is Andrew. So Andrew, you're at Columbia, is that right?
Yeah, that's right.
Okay, so you are going to be telling us about philosophy and representations, which I'm really pleased to hear about. All right, so go ahead, share away.
Yeah. Okay, so is the screen actually sharing?
Yes, it certainly is. We can see it.
Okay, good. Yeah, sorry for all the technical difficulties earlier. Right, so hi, I'm actually, I should mention from the start, I'm a bit of an outsider here. My background and my training is in philosophy, and I have some experience, though a lot less, with neuroscience. So this is all going to be from that angle, and it's probably going to look fairly different than a lot of the other talks. But what I'm interested in is something that philosophy and neuroscience share, which is a really deep interest in representation. So I'm just going to quote here from a preprint from the Cording lab. They say typical systems neuroscience experiments correlate the activity of recorded neurons with controlled variations in stimuli or behavior. Such experiments are commonly taken to reveal that neural activity is a representation of the stimulus or behavior. But what does this statement mean? Right, so we say all kinds of things. The hippocampus represents spatial locations. The FFA represents faces. Retinal ganglion cells represent light values and movement direction, and so on. What does this mean? Why is it important? And is representation essential to neuroscience? There are a lot more concrete, sort of on the ground questions you'll come across. In neuroscience, you ask tons of questions about which neural activity represents, what it represents. Does the FFA represent faces or just objects in your expert domain of discrimination, for example? What computations are performed on the representations? How are the representations implemented? How do we discover any of this experimentally? Philosophers have take a sort of 30,000-foot approach to this. So they're interested in very similar questions. Here's a quote from a very early book by Bob Cummins. He says, "The notion of neural representation is a kind of explanatory loan. If it turns out that the notion of mental and neural representation cannot be given a satisfactory explication, if in particular no account of the nature of the representation relation can be given that is consistent with the empirical theory that assumes it, then at least in this respect, that empirical theory must be regarded as ill-founded." So I think just given the success of much neuroscience that uses the notion of representation, we don't have to worry too much about it being ill-founded. But we do have to wonder what exactly the representation relation is. There's a bit of a mystery there. So philosophers have traditionally asked just one question: what is neural representation? And they have given a huge variety of answers. The representation defined in terms just of correlation, or in terms of complex causal relationships, or in terms of information carrying or information decoding, or even in terms of evolutionary selection and design. But the answers are all supposed to shed light on these questions, right? If we know what neural representation is, then we'll know which neural activity represents. It's just which neural activity satisfies the definition of representation. And we'll know what it represents because we'll know which environmental entities it satisfies that definition with respect to. So the problem is that despite there being a huge proliferation of answers to this question, there's been very little progress. There's been very little interaction between philosophy and neuroscience on the question, and philosophers have said very few things that neuroscientists have felt compelled to take seriously. And I don't think that's on the neuroscientists. I think it's on the philosophers, because I think they're asking the wrong question. And so you do see the similar questions in neuroscience as well. The paper I cited at the beginning, and in some similar work, neuroscience is very interested in defining representation. But I want to change gears and talk instead about what the notion of representation does for neuroscience. So leave aside what representation is, what that relation is, and just think about neuroscience as a science and what using the notion of representation does for neuroscience. And what I want to say is that it provides an intuitive way to project the structure of one domain onto another as a model. So I'll explain what I mean by that. Start with a sort of simple example. Just take some straightforward computer gates, or actually think of a simple circuit composed of gates like this that does something like addition. We're going to think of it in representation terms. We're certainly going to say things like the output of the circuit represents the sum of the inputs to the circuit, since it's doing addition. So again, the question isn't what is it for the output to represent something, but what does it do for us to describe it this way? And one thing you're doing is saying that the relationship between input and output is accurately modeled by the addition function, right? And I think this is generally what representational notions do. They point us to a domain of models rather than to a property that the brain has that relates it to the environment in some way. So when we say that the output of the circuit represents the sum of the inputs, we're saying that the addition function is a good model of the relationship between the input and output. And then we can begin to describe the system literally in terms of the models. So we'll say if you input, give it 5 and 7 as inputs, it'll output 12 or something like that, right? We're not describing the system in terms of its own properties, its physical or electrical properties or mechanical properties. We're describing it in terms of, literally in terms of the model that we're thinking of it as representing. So of course, you don't do this just for the input-output structure. You also do it for the internal causal structure. And so you think of an algorithm that computes or implements the addition function, and some such algorithm is going to be your model of the internal structure of the circuit. And you'll do the same thing as you did with the inputs and outputs. You'll start describing the internal structure in terms of that model. You'll start saying, this is the...
Andrew, I hate to cut in here, but I think something is brushing a mic on your end because we're getting a lot of mic sound.
Oh, sorry. Yeah. Is it still going?
No, now it's good.
Okay, good. Yeah, I think that was me. Right, sorry. So we're going to describe the internal structure of the system in terms of the algorithm or whatever that we're modeling it with. And this has obvious benefits, right? It has all the benefits of a description of a computer in mathematical rather than electrical terms. But the notion of representation specifically is helping us identify possible models, right? That the algorithms that compute the function that we've described the system in terms of. It gives us a way of linking the internal structure of the system to the model by connecting certain parts of the components, certain parts of the system to the components of the model that we think of them as representing. And it ensures that our model connects very tightly to the thing we're trying to explain, right? We're interested in the computer's mathematical abilities, so we describe it, understand it, start to finish through the mathematical structures that model it. Okay, so I should note that this is not just going to be the case with mathematical structures. Take an actual case of a computer that had to be reverse engineered, the Antikythera Mechanism, which is a computer that was discovered off the coast of Antikythera, an ancient Greek computing device for calculating astronomical relationships, discovered last century. And it's been understood in the entire history of its investigation. It's been understood in terms of in representational terms as representing astronomical entities, representing the distance between the sun and the moon, or representing the speed of the sun relative to the speed of the moon, or something like that. And so here it's not a mathematical domain. Mathematics is going to be involved, of course, but it's not a mathematical domain that we're thinking of it as representing. We're thinking of it as representing objects in the sky, right? Actual things, astronomical entities. And it's the relationship between those entities, it's the structure in astronomical, among astronomical objects that we're thinking of as the model of the device. So when we say when we say that the device represents the location of the sun, or the distance of the sun from the moon, we're saying that it is modeled by the actual relationship, the distance relationship between the sun and the moon. And likewise for its internal components too, gear trains being described in terms of their the specific, more fine-grained relationships between the sun and the moon. So you might say that the motion of the sun is being subtracted from the motion of the moon by some gear train. Again, thinking of these gears as representing the motion of the sun and representing the motion of the moon as a way of taking structures defined over those objects, the relationship in their motions, and imposing them on the mechanism as models of its causal structure. So what's going on in both these cases when we use representational notions is that we need models of relatively complex systems. And representational notions focus us on environmental structures that might provide models, whether the environment is astronomical entities or mathematical objects, and they help us connect the system to those models. So to actually bring this back to neuroscience, I want to talk about face recognition as an example. And I'm caught up to the face recognition literature to about the early 2000s. So it's going to be a very simplistic picture and a very sort of cartoon vision of even 2000s vision science, early 2000s vision science. But we think about the brain, or about the FFA in particular, as representing faces. And we think about earlier visual areas as representing lower level visual properties. Okay, and so the question is, again, not what is it to represent a face or a visual property, but why does thinking in these terms help us understand the brain? And again, the answer is that it gives us environmental structures to model the brain processes we're interested in, the ones that support the categorization of faces. So there is a structure in the environment. There's an imperfect function from low-level visual properties to the of an object, to the category that that object belongs to, whether it's a face or a non-face. And that gives us, just like in the mathematical case where the addition function gives us a model of the input-output structure of the circuit, this gives us a simple model of the input-output structure of the brain in this particular case, or in this particular task we're interested in. And then, of course, we describe the internal processes in representational terms too. And so this very this sort of cartoon version of the flowchart describing facial recognition at the top of the slide, we describe the intervening steps between input and output as representations of shapes and objects, and representations derived from them of the parts of a face, eyes, mouth, etc., bounding contours and their spatial relationships, and so on. So in this case, our models are derived not from, just like in the Antikythera mechanism, not from mathematical entities or relationships in a mathematical domain, but relationships between parts of the environment that we're interested in relating the brain to. And just like the relationship between low-level visual properties and faces, there is a relationship between a structural relationship between low-level visual properties and the shapes or objects in a scene, and a relationship between the shapes and objects in a scene and the facial, the parts of a face, or the spatial relationships between them in a scene, if they are in the scene, and then between the face parts in a scene and their spatial relationships, and whether there is a face or not in the scene. And it's those structures that provide the finer grained model of the brain insofar as it does face recognition. So when we think of the brain as representing these finer grained features of the scene, between low-level properties and the category something belongs to as a face or not face, we're again using representational notions to pull structure from the environment, pull structures defined over environmental entities onto the brain as models of its causal structure. Okay, so that was quick, hopefully relatively clear. I'm just going to give a few upshots and then conclude. So first of all, I think we can understand representation in neuroscience entirely at the level of what using representational notions allows us to do, without asking at all what representation itself is. I think the notion of representation allows us to impose environmental structures onto the brain as models of its causal structure. And the picture is going to get more complicated as we look at more complicated visual processes and a more detailed understanding of them. But I think this is fundamentally what's going on when we talk about representation. This is extremely useful as long as the brain's causal structure actually does mirror environmental structures at a relatively fine grain. But representational thinking on this approach is just one sort of modeling resource. And in many cases, it's not going to be the most useful tool. And so to answer the original questions that I started with about what neural structures represent and so on, we really have to ask, when we ask those questions, we're really asking how we should go about modeling the neural structures. And so there are lots of implications here for debates over the relevance of behavior to neuroscience and debates over naturalistic stimuli and so on. But I will end there and thank you all for listening. And I'm very happy to answer questions, or my email's on the screen. So if you want to email comments or rebuttal or anything like that, I'd be happy to talk. Thanks.
All right. Thank you, Andrew. That was great. Always welcome to have philosophers in the computational neuroscience world. We definitely need more input and more sources of input. Do you want to stop sharing screen? I don't know if I can do that without booting you off the stage.
Yes, sorry.
If I do. There we go, perfect.
Okay. Thanks.
So in the interest of time, I think we're going to have to move on, but you can answer questions just by typing into the Ask a Question box. So it's kind of like a message board. So go ahead and do that. And once again, thank you, Andrew. All right, and now we're going to move on to the final talk of this session. Abdullahi, who is speaking to us very late. I believe it's almost 1 o'clock in the morning there.
Yes,
he's at the Donders
correct.
Institute over in the Netherlands. So please, Abdullahi, go ahead and tell us what you tell us.
Yeah. Well, sorry, just a second. Try to share the screen. All right, I think you can see it.
Yep, looks fine.
Somebody can confirm. Perfect. Do you also see my
You
cursor?
can, yep.
Okay, that's fine. Then I'll just start. All right. Yeah, thank you. Thank you all for tuning in. I'm going to talk about a project that I've been working on for a while now on the relationship between predictive coding and energy efficiency. As you may know, in neuroscience, the predictive coding framework has arisen as sort of a prominent and very powerful theory of neural information processing. It postulates that there is some hierarchical generative model in our brain that continuously inhibits predictable sensory input. And it does so. It also comes with a few sets of assumptions that, for example, that there are distinct types of cell types in the brain, such as prediction neurons and error neurons, and that these also interact with some specific connectivity motive to implement the information flow of predictive coding. So there are a lot of assumptions there, and these are also typically implemented in computational models of predictive coding, but they are generally hardwired. So a popular one you can see here on the right, it's PredNet. Some of you are probably familiar with it, but this hardwiring is typically is baked in computational models of predictive coding. So you can also ask another question. So where does this architecture exactly come from? And that is what we were interested in this work. So can we potentially explain predictive coding through other, maybe simpler mechanisms? And we think that we can do this. And in this work, we show in a specific setting that when you minimize for energy, you can get the sort of architectural hallmarks of predictive coding to emerge. So how did we do that? So there's a lot going on in this on this slide, and I unfortunately don't have a cursor or a laser pointer to get you through it, but I'll try to do my best by being as expressive as possible. So we feed in sequences of MNIST digits in ascending order. So what does that mean? If you look on the left, you see 6 as the starting digit of the sequence. So after a 6 comes a 7, then an 8, then a 9, then we go to 0 because we have wraparound, etc., etc., etc. If you move then to the network, each unit in the network is connected to exactly one input pixel. And each unit is then connected to all the other units in the network. And of course, we have to make sure that the network can't just trivially turn off its input weights to achieve zero activity or energy. So we fix the input weights. If we then look at a specific unit, the output of that unit is going to be its input drive, which you see on the right in yellow, and its recurrent contributions from the units, from the other units in the network, which we denote as the internal drive. Push that through a ReLU, and then you get the output of the unit. So there is no overarching task. That is a question that I often ask. So what is the exact task that the network is supposed to solve? The only thing that we do is we push sequences of MNIST digits. Those will cause activity, and the objective is to minimize that energy. And so that essentially is the task, and there's no additional task where there has to be classification or anything else. So I've already mentioned energy, I think, a couple of times. So what do I exactly mean with that? So in this work, we take the mean absolute pre-activation. You can see the mathematical form here on the right. So in the preprint of this work, we show that this loss correlates pretty well with action potentials and synaptic transmission, which are often noted as the 2 biggest sources of metabolic demands in the brain. And you can see a nice pie chart here on the left to drive that point home. So the first thing that we have to check is whether the network is able to predict its incoming pixel activity. And since the network has a nice topographical correspondence with the input, recall that the each unit is connected to exactly one input pixel, we can easily visualize that in the MNIST case. And you can see this visualization here on the slide. So excitatory, so the red here means excitatory and the blue here means inhibitory. And as you can see, the internal drive here is able to counteract the input drive by not exactly mimicking the shape of the input, but by predicting what is essentially a median digit. And this median digit, which is essentially the pixel-wise median for that digit class, is the sort of optimal solution given this loss function. In addition to that, you might notice that there are some red dots in the internal drive, and these are units that have positive internal drive, which is interesting. Could this potentially be distinct populations, and could these essentially two distinct populations be the functionally correspond to the prediction and error units that that are sort of postulated in the predictive coding framework? That is what we try to find out next. So if the network would reorganize into two distinct populations of prediction error units, we would expect them to have different properties. And in particular, in this setting, we would expect them to have differences in their median pre-activations. So the reasoning behind this is that if, say that we have a unit that has a bias away from zero pre-activation, well, it essentially must mean that this unit has a functional role in driving down the activity of the network. Since if that wouldn't be the case, the network would just turn off that unit because to reduce the loss. So we use that to identify candidate prediction units. So what we did is we recorded for each class the median pre-activation and constructed confidence intervals. And then we made a decision rule that essentially says if for a particular class that unit's 99% confidence interval does not cross zero, then we identify it as a prediction unit. And here on the slide, you can see an example of that for the digit class 9. So on the right, you can see a decision boundary, and you see that two units have crossed the decision boundary. Up until now, I only have shown you how to identify these units, but I haven't shown you, I haven't demonstrated to you that these units actually play a causal role in the prediction in the network. So that's what we try to figure out next. So to figure out whether these candidate prediction units actually play a causal role in prediction, we did a virtual lesion study. On the left side, you can see a visualization of that. So what you see here is we visualize the energy consumption, which is the pre-activation over time. And each line here essentially denotes a multiple network instances for bootstrapping, and they are averaged over multiple sequence presentations. So we have three situations here. The red line here denotes the normal operating, normally operating networks, and the green line is a control where we lesion a randomly selected set of what we then call error units, postulated error units, to make sure that the effects that we might see in a lesion is unique to the prediction units. So we're going to focus first on those, on the red line and the green line. As you can see, these networks are able to reduce their energy consumption over time, meaning that they are able to functionally, essentially predict their input, because the only way that you can reduce the energy consumption in this problem is by being able to anticipate what your input drive is going to be. which also means that lesioning these error units, postulated error units, doesn't seem to have a effect or a deterioration on the prediction. So you also see here a yellow line, which represents the case where we lesion the candidate prediction units. And here you see now that the network isn't able to reduce its energy consumption over time as well, compared to the other two cases that we just discussed, which implies that these candidate units seem to play a causal role in the prediction. In panel B, you see the same thing, but represented in a different way. So if we look at the top row, you can see predictions of the digit class 0 at different times in the sequence. And as you would expect, the further along in the sequence, the better the prediction is, because the network has seen more of the sequence. On the bottom row, you can see a visualization with a network with lesion prediction units. And there you can see that the predictions don't improve, but they also are not completely destroyed. So this implies that there might be two different mechanisms of predictions maybe going on. One faster predictive inhibitory loop within error units when the prediction units haven't had time to integrate information yet, and a slower predictive loop that is mediated by prediction units. So to sum up this slides, knocking out these prediction units deteriorates the predictions, but they are not completely destroyed. To add to these results, it is also important to want to have some form of replication, since as you've already seen, the MNIST input is pretty particular, with drive in the center and no drive in the periphery. So we also looked at C410, and we saw a similar phenomena unfold in that data set as well. So again, as you can see in panel B, we can sort of identify these, let's say, candidate predictive units, and lesioning these prediction units, as you can see in C, leads to increased network activity, which implies that they have some functional role again in prediction. So to sum up, we are able to, via energy efficiency, to sort of recover hallmarks of the predictive coding architecture, such as functionally distinct populations, and even maybe some form of rudimentary hierarchy. In addition to that, these populations functionally map well on prediction and error units, which is nice to see. So in this instance, we can somewhat conclude that predictive coding, or at least sort of the entire architectural baggage that comes with predictive coding, might be a consequence of energy efficiency. If you're interested in this work, we have a preprint of this work on bioRxiv. You can scan the QR code here on the right. And thank you for listening. I also want to thank my collaborators. And if you want to contact me and discuss me after this, I'm on Twitter, and you can always send me an email. And I'm also happy to take questions. Thank you.
Great, thanks. That was great. Can you go ahead and unshare the screen? Oh no, actually, yeah, there you go. Perfect. So we got a few minutes before the next session begins. Let me get to, first of all, Kyle Johnson's question, who asks, these prediction units had receptive fields, but just in unimportant areas?
So do you mean, yeah, so in the MNIST case, the prediction units all emerge in the periphery where there is no input drive. So yeah, there were receptive fields there with respect to the input is going to be meaningless, but that also makes them stable, which also might be the reason why the network recruits these units to become the postulating predictive units. Yeah.
I had another question, which was that it would seem as if the loss function was just trying to minimize energy, wouldn't it predict that basically it would drive all the synapses to zero?
Yeah. So
is there something to do to sort of prevent that from being a trivial?
Yeah. So that would happen if the input weight would be plastic.
Okay.
So we fixed that to make sure, because so the problem statement is essentially there's going to be drive, and you need to be able to adequately deal with that drive.
Mm-hmm.
And for that, we have to freeze those weights. But in essence, I feel like this is a situation that translates, I guess, to a circuit in the brain, because we can't just close our eyes and sit somewhere. We have to actually engage with the information stream. So in that essence, I think this is okay. But yeah, it's sort of a hard code trick to deal with that. Yeah.
Right. And I can see how that would work for a one-layer deep system. But then I wonder if you had multiple layers, right? You can't force these guys. You can't force everything up to this current layer to be fixed. Like they've all got to be plastic all the way down the line.
Yeah. That's true. And that definitely would be the next step. So in essence, at some point you have to. So the problem is, I think the solution there would be, if these higher order layers can learn something useful that you wouldn't get if you just have a shallow network, then naturally the network would recruit them. So it's all about like a trade-off, right? So how much does it cost to have these circuits to be active, and how much do they actually give you to solve the problem?
Right.
So it will probably be something like that. That's definitely
some kind of value function for what's the utility of these weights not being zero.
Yeah.
Right.
Like if they're super expensive and they don't give you any like functional gain, then obviously you don't want them to be on. But if like having this like additional latent structure gives you like a better solution to, for example, minimize energy, then the network will probably opt into using those structures. Yeah.
All right. Well, we are out of time here. Thank you very much for those 3 very interesting talks. If you guys just hang tight, we're going to get pulled along into the next session and hear the last 3 talks of the NeuroMatch session. So thank you one and all. Really appreciate it.
All right, welcome everybody to the last short talk session and the last session of the NeuroMatch conference, NeuroMatch 4. We've got 3 wonderful speakers for you, and then stick around at the very end. We'll have some fun stats for you to kind of close out and round out the conference. So for those who don't know me, my name is Megan Peters. I'm a cognitive neuroscientist at the University of California, Irvine. And today it is my great pleasure to host our last short talk session. So today we have Tom Burns, Guangyao Qi, and Samira Glazer Khan. I'm sorry if I butchered your name. I think we will just go ahead and get started with the first short talk. So Tom, do you want to take it away?
Sure, thank you very much.
Okay, fantastic. Yep, I'll hit the screen share button.
Yep.
Looks beautiful.
Okay, yeah, thank you very much.
All right, here we go.
Click hide. Thank you very much, and thanks for the hosting. And yeah, it's been a great conference so far. I'm quite honored to be in the last session. So my talk today is about multi-scale and extended retrieval of memory structures in a model of local global inhibition balance. And this is joint work with Tatsuya Haga and Tomoki Fukai at OIST in Japan. So we typically think that there are assemblies of excitatory neurons in the brain. And for example, here's a picture of different electrodes placed in CA1 of hippocampus. And we see that there are clusters of excitatory neurons being activated synchronously over a fairly large distance. So these are sparse and distributed assemblies. What's more is we see that these assemblies actually happen relatively close to the troughs of theta waves. Theta waves are a particular band of frequencies within the local field potential, the local electrical current in the brain. And in hippocampus, these assemblies preferentially occur or tend to occur in these troughs or particular phases of the theta wave, the theta frequency of the local field potential. And the theta wave is largely determined by, or at least influenced by, inhibitory neurons, although there are other influences. So we're wondering what are these inhibitory neurons doing other than maybe just sort of synchronizing these activities. We're also wondering, how does the association between assemblies get influenced by inhibition? So if we have one assembly here of excitatory neurons, let's say representing a lion, we might see that some of these neurons in the same assembly also represent, let's say, a leopard or a tiger. And the extent of overlap between these assemblies, or the neurons that make up those assemblies, we could think of as the degree of closeness between these two memory items. So these assemblies we can think of as a memory item, and how much they share or how much they have in common in terms of their neurons or connections, we can think of as their association or degree of association. But as I sort of referenced regarding the theta wave, what are the inhibitory neurons doing in all this? Well, a classic view is that there's a blanket of inhibition. So for example, we have some excitatory current onto some individual neuron, which is up here. And then we have some inhibitory current, which sort of cancels it out so that we have a net current here. This dashed line is the threshold for this neuron. And we can see that if we had only excitatory current coming to the cell, the spike train would be just constant for this neuron. But we don't want that. We want something which is actually giving us a signal. And so we balance it out with inhibition, and we get some net where it only broaches the threshold at certain points, and we only get spikes at certain times. So that's the classic view, that basically there's a balance overall between excitation and inhibition. But the emerging view is that there's a computational role as well. So there's many different lines of evidence for this, but I'll just point out one, and that is that if you look at pairs of neurons in close proximity, one excitatory and one inhibitory, you'll find that instantaneously, that is, there's no chance for any sort of feedback or interaction between these neurons, instantaneously they will exhibit some tuning which is similar. So for example, this excitatory and inhibitory neuron are experiencing the same excitatory postsynaptic potentials. Well, I wouldn't say exactly the same, but they're sort of similar in time and magnitude. Oh, and you can also find opposite relationships where there's an inverse correlation between the inhibitory and excitatory neuron in terms of their postsynaptic potentials. So this implies that the inhibitory neurons are also tuned, like the excitatory neurons, to particular stimuli. So they have potentially a more complicated role than simply balancing out the excitation. So we made a model, which we call a cortical model, but it's still quite abstract, where we have two types of inhibition, global and local. The local are tuned and the global are not tuned. We start with a graph, essentially, of excitatory assemblies. So each one of these circles represents an excitatory assembly. And we have some, if we just focus in on one assembly, then we have some number of neurons in this assembly, which are all-to-all connected. And then we associate them through a connection to their neighbors, so to their associated sort of memory items, let's say, but they're not connected to everything else. They're only connected locally to things that they are associated with. And then they also, all of the excitatory assemblies also drive some sort of global inhibitory population, which also has a certain number of neurons in it. Here we have 500. And that globally sort of controls and gives some sort of excitatory inhibitory balance. But then we also have these local inhibitory neural populations, which are paired with each excitatory assembly. So we have local EI excitatory inhibitory assemblies, which for each memory item. So I've only drawn it here for one pattern or one item, but actually it's for all others as well. And then what we do is we put in a little bit of excitatory stimulus to one particular assembly, and then we ask, we turn it off, and then we ask what happens to the activity of the network? How much is the activity spread in terms of excitation and correlation between the activity of the different excitatory ensembles? And finally, what we do is we have a parameter called C, which we vary between 0 and 1. At 0, the local inhibitory synapses are essentially 0. They have no influence on the excitatory ensembles. And at 1, it's the opposite. The global inhibitory population has synapses which are essentially 0, going to all of the excitatory assemblies. And so with this parameter C, we can balance between a globally and locally dominant inhibition structure. And at 0.5, we're exactly in the middle. And this network has about 4,000 excitatory neurons, not in each assembly, but divided across all the assemblies. And the typical joint assembly size is about 40 excitatory neurons and 10 inhibitory local neurons. And then we have this group of 500 inhibitory global neurons. And we also have some response functions here. This is a rate model. So you can see the preprint for further technical details. Now, what we decided to do is we started with a very simple structure. So these excitatory neurons are connected together in a graph. And we started with a 1D ring. So we have here just an illustration of the activity. The darker the vertex is, the more that assembly is active. And we started at this particular vertex that this arrow is pointing to. And you can see that the activity gradually decreases as we go around the ring. And that indicates that the activity spread to those vertices during the simulation, but didn't go all the way around. And it stayed within, let's say, in this case, like five distance away, 5 vertices away. And if you look at B, you can see that we tried this with different values of C. And on this x-axis is just that distance that I was sort of like hand waving about on A. And you can see that when it's totally global, that is at C equals 0, the distance is, let's say, 5 or something. But as you increase it up, let's say to 6, 0.6, you get a distance of maybe 10 or something. And what's interesting, though, if you increase this just ever so slightly more to 0.7, then you find that it's very noisy. Each one of these gray lines is a different trial. The thick black line is the average. And you can see that there's a trend, but it's extremely noisy. These are the correlations of activities between different vertices activities, different assemblies. But if we increase the size of the assemblies and the network, twice, and then twice again, and then twice, which is finally 8 times larger than the original, we get rid of this noise and we get a nice, clean-looking correlation structure, which extends the association up to about 20. So this implies that maybe these sparse excitatory inhibitory joint assemblies are required to be of a certain size in order to maintain this effect and this extension effect over a certain distance. But nevertheless, we still see that starting from a global situation at c equals 0 up to 0.7, we actually quadruple the range of the retrieval. And that's quite interesting because we haven't changed anything about the excitatory structure at all. We've just changed the balance of activity or strength of activity between 2 populations of inhibitory neurons. We also did this with more sophisticated graph structures. So you can see here, if on the left column, we have c equals 0.1, and on the right column, c equals 0.525. And you can see in general, at the higher level of c, we spread out the activity more. And here on the right in B, we've got different correlations, correlation matrices. The darker the pixel, the more correlation there is, and the lighter it's a negative correlation. And you can see that for different structures or for different graphs, you can pick out different structures or different sort of clustering effects. And I won't go into all the details here. I encourage you to look at the paper, but it's very interesting if you look quite closely at some of these, that it's picking out very fine topological features at some levels, whereas it's looking more at global features at others. One particular example that fascinated me was the Tut graph. And the Tut graph is quite special because it has some interesting symmetries. Like, for example, it's 3 regular, meaning each vertex has 3 neighbors and only 3 neighbors. But it also doesn't have any path starting from a vertex and coming back to that vertex without crossing over extra paths. So it has this asymmetry, but also a strong symmetry. And that makes it interesting to pick this central vertex, because from this central vertex, if you activate that, you can go into 3 rooms, but which room do you choose to go into or become more associated with? How much do you spread the activity into those 3 different rooms is a bit like maybe how much do you explore or exploit different parts of the environment. And you can see in the more globally dominant case, we start in the initial part of the simulation, so this is at 61 to 80 milliseconds, we start exploring all 3 rooms, but quickly it goes into this winner takes all dynamic, and one of the rooms wins, and eventually that becomes strongly activated at the end of 500 milliseconds at the end of the simulation. Whereas when it's more locally dominant, we spread out the activity more at the beginning, and we actually maintain this all the way through, which is sort of like a winners share all dynamic. And so we speculate that there's this switching between local and global inhibitory activity scenarios, which might be actually useful behaviorally, for example, in exploiting or exploration. We also did some calculations on how much the activities in the model are relating to clustering, so topological features, or just pure geometric spread in these abstract graphs or arbitrary graphs. So that is a geometric value. And the closer these values are to 1, the more information is similar between the activities that we found in our simulation and the activities you would expect if it was purely looking at, say, topological information on this left column or geometric information on this right column. And you can see that in general, there's a mixture between both of these pieces of information, and it changes depending on what value of C you pick and what particular graph you're looking at. So in summary, local inhibition dominant states can extend the recall of sparse assemblies by 4-fold in a 1D ring memory structure. And local global inhibition balance varies the scale of topological and geometric focus on activity in sophisticated memory structures. The preprint for this paper is out now on bioRxiv. You can scan this QR code to look at it. You can also look at my Twitter, and also I'm job hunting in next year. So please contact me if you're interested. Thank you very much to my funders and co-authors and to Neuromatch. And thank you for your time.
I will applaud on behalf of all of the audience. Thank you very much for this really interesting talk. We have time for at least one question here. So Chris Roselle is asking, does the type of inhibitory strategy, local versus global, impact how many inhibitory neurons are required? Wondering if one is more consistent with observed EI cell type ratios.
So overall, the model maintains the same EI ratio in both global and local scenarios. And in general, I suppose you could say that, of course, there are more than 2 types of inhibitory types or connection types. So I would say that, yeah, potentially this balance is also balanced with other needs of inhibition or other computational demands. And so, but in general, yeah, the model maintains the same EI balance of about 4-to-1 or 5-to-1.
Great, thanks. And one more quick question here. Emmanuel Tognoli, oh, I'm sorry, I butchered that name, is asking, does winner share all network with greater global coupling exhibit slower dynamics? Are there invariants across temporal scales, enlarging the time scale and global coupling case accordingly?
So we didn't try different temporal scales in terms of the internal dynamics of the neurons, but you did observe there in one of the slides that the temporal dynamics of the change to a winner takes all scenario is quite quick compared to the winner shares all. So if you look, it's not shown in that particular plot, but the winner share all is a bit slower, and on the peripheries there's this sort of democratic debate about which one is going to win or something like that. So you can maybe see that in the supplementary of the paper.
Fantastic. Thanks.
Yeah. Thank you.
All right. If there are no more questions then right now, please also feel free to use the Discord channel to discuss this. As Brad said in the last session, that Discord server is not going away at the end of the conference, so please continue the conversation offline. Thanks very much, Tom. So our next speaker is Guangyao Qi. And unfortunately, Guangyao's internet is not doing so hot right now, but what we're going to do is play a pre-recorded talk, and then Guangyao is here to answer any questions that you might have about that. All right, so here is me attempting to share my screen, and I will do this, and I will share the tab audio, and hopefully what is happening now is that you all can see and hear this. So I'm going to hit the play button and hope that this works. Here we go.
Hello everyone, I'm Guangqi. Today I share a story about how the macaque brain solved the causal inference problem during multisensory processing. We are living in the multisensory environment, and to create a coherent perception of the world, the brain has to combine noisy information from multisensory cues. And the causal inference is the process to determine whether multisensory cues come from the common cause or different causes. For example, when a mosquito bites you and you want to swat it, basically we received the proprioceptive signal and the visual signal. If a common cause is inferred, you will integrate these 2 signals and swat here. Otherwise, different causes are inferred, and you will ignore the proprioceptive signal. And the key of the causal inference is to infer the hidden causal structure of the environment. How to solve this problem? Previous studies show that humans infer the causal structure by Bayesian inference, which indicate the sensory evidence and the prior information to get the posterior probability for optimal inference. And for Bayesian causal inference model, it assumed 2 causal structures. And during the reference inference processing, it inferred the uncertainty about the causal structure. That is the posterior probability of common source. And neuroimaging studies show that the progression of causal inference commutations along the cortical hierarchy. However, few was known about it at the neurocircuitry level, and how the brain represent and update the hidden causal structure is largely unknown. Here, we asked the two key questions. And to address these questions, we conducted single unit recordings in macaques while they performed a causal inference task. During this task, the monkey was inferred to reach the target by his perceived arm, and we manipulate the visual arm to manipulate the disparity. Here, when we change the disparity, the inferred causal structure was changed. And the monkey had to
Sorry, folks. Technical difficulties. Give me a second. The video will not play. It seems the technical gods are against us right now. Please bear with us. Oh, dear. All right, we are at 3:10. I'm going to try reloading the page.
Hello, everyone. On the inferred causal structure. Importantly, here we define the disparity as the real arm to the visual arm. And since we asked the monkey to reach the target by its estimated arm, so the drift could be defined as the real arm to the visual arm, the target. And here is an example trial. This condition is called VPC condition. And besides VPC condition, we have two control conditions, the VP condition and the P condition for integration and segregation respectively. We recorded single unit in the parietal and premotor cortex, since this brain area is highly related with the multisensory processing of body representation. To examine whether the monkey inferred the hidden causal structure, we first test the drift as a function of disparity. As we can see, when the disparity is 0, the drift is around 0, and with the increasing of disparity, the drift firstly increases almost linearly. Then the increasing rate slows down and even has a decreasing tendency. We use a Bayesian causal inference model to capture this behavior. And we found that causal inference model could capture this nonlinear dependence. And the monkey used the model average strategy for final estimation. And more importantly, the inferred causal structure, that is the posterior probability of common source, the P common, is decreased as the disparity increased. And to investigate whether single unit could represent a causal inference, we calculate the normalized integration probability for single unit. And if the repeat is correlated with P common, we define this unit as a causal inference neuron. And the fraction of causal inference neuron in premotor is higher than that in parietal cortex, and which indicates that the promoter could play a different role in causal inference processing relative to parietal cortex. And to test it, we first conduct the population decoding analysis. As we can see, both premotor and parietal cortex could decode the peak common, but only premotor cortex could decode the peak common during the preparation period. And with GPCC analysis, we found that there is a communication of peak common from premotor to parietal. As we can see, here is a it shows that the correlation between the PMC and IL-5 is unbalanced, and the premotor leads IL-5. For now, we found that the causal inference neurons are distributed in premotor and parietal cortexes, and only the premotor cortex could decode the peak common during the preparation period, and may send top-down modulation to the parietal cortex. So you ask the next question. So how the brain update the hidden causal structure? We hypothesize that during the causal inference processing, the monkey will update the prior of common source, and to match the causal inference environment, it will update the sensory representation. So we focus on two parts. One is the prior distribution of common source, and the other one is proprioceptive representation. And to investigate whether historical information could influence kind trials prior, we adapted a marker processing for sequential trials. And here, as we can see, the transition probability between common source to common source and separate source to common source is unbalanced. And at single neuron level, we found that premotor neuron is selective to prior information during the reference period. And at population level, we found that only premotor cortex could decode prior information during the reference period. Finally, we found that there is a trade-off between inferred causal structure and sensory uncertainty. Here shows an example session. As we can see, the uncertainty of proprioceptive representation in VPC condition is lower than that in VP condition. And overall, the monkey's behavior shows that the uncertainty in VPC condition is lower than that in VP condition. And at the single neuron level, we found that the tuning curve of VPC condition is disturbed relative to VP condition, and only parietal cortex could update the sensory representation. To summarize, we found that the monkeys, first, monkey's behavior show that it integrated the prior information and current sensory information to infer the causal structure, and then it will update the prior of causal structure and update the sensory representation to match its causal environment. And at the neurocircuitry level, we found that a premotor cortex updates the prior information of causal structure and represent the co-computation of causal inference, and send top-down modulation to parietal cortex for sensory updating. And finally, I want to thank my advisor and my collaborators, and thanks for your attention.
Great, thank you. And I'm so glad that our technology worked and we were able to make that work. So I appreciate everyone's patience while we got our technology to work. And thank you, Guangyao, for preparing the talk ahead of time to make that work. So we don't have any questions in the Ask a Question button, but I have a couple. So you're showing that this kind of the transition probabilities from probability that C equals 1 to probability that C equals 1 is higher than going from probability C equals 2 to C equals 1, right? Did I understand that correctly? Yeah. So, so I'm wondering if you've looked at this in the context of any kind of one-shot learning or serial dependence effects in your actual stimulus presentation, just because Dave Wozni and Brian Odegaard have shown some really interesting serial dependence one-shot effects in these causal inference structures. So have you looked at whether if that difference in transition probabilities is even stronger when you have that sort of serial dependence in your trial structure?
Yeah, I think probably, but in our task, the trial is independent. Trial by trial is independent, so the prior information is very weak. Yes.
Right, but there might be some serial dependence that shows up even though the trials are independent. Oh, can you still hear me?
Yeah.
Okay, so there might be some dependence that shows up even though the trials are independent, like pseudo-randomized. There might still be some kind of unintentional serial dependence that shows up, right?
Yeah, but we didn't test that, but we tested at the neural level. When you come back to two trials, it won't decode the information in parietal cortex. In premotor cortex. So I think the information will decay sharply.
Great. I'm sure we can talk for longer, but we should probably move on to our last speaker. So again, if you guys have any questions, please feel free to use the Ask a Question button or the Discord server for offline communication. So thanks, Guangyao, for being with us, and we will now move on to our last speaker of the session. And so, Samira, are you here? I'm ready to go.
Yes.
Great. Do you want to turn on your camera or do you want to stay off?
Yeah, I will turn it on.
Okay. Fantastic. Okay, and so we'll share screen and then we'll get going.
I'm having a little bit of trouble with the, here it is.
The menu hides until you hover over yourself.
Okay.
There we go. Looks beautiful. All right, take us away.
Wonderful. Okay. Hi, everyone. My name is Samita, and I'm a student at Yale in the K-Lab. And today I will be presenting our results from 2-photon imaging of norepinephrine and cell firing in the mouse prefrontal cortex. So norepinephrine is an arousal-related neuromodulator, which is released from neurons of the locus coeruleus that project up to higher brain structures and that have their nucleus in the brainstem. And norepinephrine is released via volume transmission, which means that it's released extrasynaptically via vesicle exocytosis on different locations of the locus coeruleus axons. And previous research showed that norepinephrine is released homogeneously within the prefrontal cortex. But recent studies have been showing that norepinephrine release can be heterogeneous and spatially localized. So we are interested in studying spatially localized norepinephrine release because it has important implications for different behaviors, such as selective gain of attention. So this study by Mather and Kliwit in 2016 showed that during arousal, norepinephrine can lead to spatially localized hotspots where neurons are especially active, and that those are involved in attention to high salience targets. So our hypothesis is that norepinephrine is released heterogeneously across the prefrontal cortex. And we think that the mechanism that's underlying this heterogeneity is differential recruitments of different subsets of norepinephrine release and reuptake sites over time. So the method that we use to get at our hypothesis is two-photon imaging. This data set was collected by Neil Savalia in the Quan lab at Yale. And so basically the way that this works is we use two constructs to infect the mouse's prefrontal cortex. And we have one red calcium sensor and one green norepinephrine sensor. And then we have our two-photon microscope come in and excite both of our fluorophores. And then we're able to gather data from both our red channel and our green channel. And as a result, we'll have fluorescent recordings of both norepinephrine and calcium activity. And then we're able to go into these recordings and extract fluorescence time series from locations that we're especially interested in. And for the analysis of these time series, we use primarily generalized linear modeling and optical flow. So first we were interested in looking at the local norepinephrine field compared to the global norepinephrine field. We define the local norepinephrine field as the norepinephrine fluctuations within a radius of about 15 microns of the cell. And the global norepinephrine fluctuations are all of the remaining norepinephrine fluctuations. So here on the right, you see two time series. At the top, we have the fluorescent fluctuations for global norepinephrine, and at the bottom, we have the ones for local norepinephrine. And we observed that there were certain time intervals where these two time series oppose each other, leading to a decreased correlation between local and the global norepinephrine field, which points to heterogeneity. And next, we were interested in looking at the mechanism of this heterogeneity. And to look at the mechanism, we wanted to observe norepinephrine release and reuptake. And in order to do that, we used a method called optical flow, which tracks small-scale phasic norepinephrine fluctuations. This is an example of a pseudo-colored recording of norepinephrine fluctuations. Anytime you see a region of dark blue, that's where there's an especially low concentration of norepinephrine, whereas yellow is a high concentration. And you can see that these vectors track small-scale fluctuations. So using these movies, we're able to extract the time and location of norepinephrine release and reuptake. And we observed that norepinephrine release can occur at the same sites, but at different times. And this is depicted in the panels on the left. And statistically, we were able to verify this observation by showing that release and reuptake are spatially highly correlated, but temporally anticorrelated, which indicates that release and reuptake occur at the same sites, but at different times. So next, we were interested in examining the relationship between local norepinephrine and cell firing. In order to get at this question, we used generalized linear modeling. We fitted models predicting cell firing, and we used local norepinephrine and global norepinephrine as predictors. In the schematic in panel A, you can see the local norepinephrine is again the radius of 15 microns around the cell, whereas the global norepinephrine is the remaining fluctuations. And within our model, we also accounted for the relationship between local and global norepinephrine, because there are times when these two time series are correlated. And we fitted these models to 83 different cells, and what we found is that local norepinephrine positively predicts cell firing, whereas global norepinephrine does not. Next, we were interested to see what would happen if we broke the relationship between local norepinephrine and cell firing. So we hypothesized that this relationship is dependent on these small-scale norepinephrine fluctuations that were visible in the optical flow movie. And we wanted to remove those, and we were able to do that using a drug called desipramine, which blocks norepinephrine reuptake and causes the accumulation of norepinephrine in the brain, which subsequently takes away those small-scale phasic fluctuations. So when we repeat the modeling procedure under the influence of desipramine, we see that local norepinephrine is no longer a significantly positive predictor of cell firing, which confirms that this relationship is dependent on those small-scale phasic fluctuations. So in summary, what we're able to conclude is that norepinephrine is released locally, that local norepinephrine affects cell firing, and that the effects of local norepinephrine on cell firing are dependent on small-scale phasic fluctuations, and that this relationship can be broken through the blocking of norepinephrine reuptake. I'd like to thank everybody in my lab, especially my mentor, Alfred Kay, and also thank you to everybody else who worked on this project. In the Quan lab, we have also Neil Savalia and Gianna Cressy. Thank you so much.
Well, I will clap for everybody. Thank you very much, Samira. This was really interesting. We actually have a lot of time for questions. So I will kick us off with a question, actually. So you're recording from PFC,
Yes.
Is that right? No. Where were you recording? Yes, PFC. So do you feel like this relationship between phasic dependent local norepinephrine release and the firing rate of neurons is unique to the PFC, or is this kind of a general rule that we might find everywhere in the brain? Do you have any thoughts on that?
Yeah, for sure. So I think that there's been previous research that shows that the norepinephrine transporter is expressed in different levels and different layers of the brain and in different regions. And I think that the PFC, or especially the region that we recorded, is a region where there's a relatively low density of those norepinephrine transporters, which kind of allows for these pinpoint release and reuptake sites to dominate the fluctuations. And so I think that in different brain regions, depending on the expression of this transporter, there might be different fluctuations that we observe.
Great, thanks. We actually have another question already. Brad, who is one of our NeuroMatch organizers, Brad Weibel, is asking, do you think the local fluctuations are driven by local activity, or are they driven by norepinephrine heterogeneity?
Could I clarify? Do you mean like local, if they're driven by local cell activity, or if it's driven by the norepinephrine field?
Oh, that's a good question. Brad, can you type in the chat what your local cell activity?
Oh, okay. That's a really good question. So I'm actually not sure. It would be really interesting to see how the relationship is in reverse. So I would be really interested to see how cell firing affects the local field. The way that we did it and kind of like the direction that we were looking at is that this heterogeneous field is just kind of created by heterogeneous release, and then that acts on the cells. But I'm sure that the relationship is somehow also reciprocal, and that cellular activity influences the norepinephrine field as well.
Fantastic, thank you. We have another question here from Ning, who is asking, why was 15 microns chosen as the criteria for local norepinephrine activity? Have you looked at other radii?
Oh yeah, that's a really good question. So we chose 15 microns because we actually looked at basically every other radius up to around 100 microns. And we found that the significance of the local field drops off at around 30 microns. So 15 was just a happy kind of medium within that region. But yeah, we definitely have looked at other radii. And what we found is that basically at 30 microns, there's this really sharp drop off. And beyond that, it's no longer significant. But up until 30 microns, basically any radius that you use gives the exact same results.
Very interesting. Thank you. All right, let's do one more. if we've got stamina. I know it's the end of the conference and you're sending us off with a bang. This is awesome. So last question from Emanuele Tognoli is asking, anti-correlated is a strong dependency. Time series 1 strongly predicts time series 2. Plain correlations do not make justice to them. Have you considered metrics and theoretical concepts like bistability and metastability?
Stability and metastability. I cannot say that I have. Yeah, no, we were looking at very like simple correlations, but I would definitely love to go back and maybe try something more sophisticated on that, because I do think that the relationship between local and global norepinephrine is very sophisticated. But yeah, for now, we were just doing simple correlations.
Fantastic. Well, I hope that you guys then can continue the conversation offline. So as a reminder, our Discord server will stay open for the foreseeable future. So I want to thank all of our speakers for this last session. This was fantastic. Thank you to everybody who was here for the NeuroMatch conference. This was fantastic. So don't log off just yet. I'm going to take like 3 seconds to pull a number of other people here onto the screen who are part of the NeuroMatch conference. So if you give me just 1 second here to do this. Okay, here's Titty Pat. Fantastic. Brad, are you logged in as yourself or are you NeuroMatch? Can you be yours? There we go. You are logged in as yourself. Okay, fantastic. So I've got Titty Pat, who is our tech wizard. All of the technical everything that you see that has to do with NeuroMatch is in no small part, and in fact, basically all of it is Titty Pat. And so that's fantastic. Oh, hang on. Brad, I think I invited a different one of you onto screen. So I think there's like 3 of you in the audience. I'm not sure. And so I've got Sarvanaz, are you here? Do you want to come on screen if you want to say something? There's Brad.
Hey, Brad.
Want to say something as well. And let's see who else we can get onto the screen. Who else is here? Chris, I prompted you. Can you see this? Who else is here?
I think Chris had to go deal with his.
Had to go. Yeah, that's right.
Yeah.
That's right. He had to go do real life NeuroMatch things. All right. I'm going to bring Sarvanaz on because she's here in the audience.
She says
no.
She says she's not at a camera.
All right. Fair enough. This is kind of a surprise here. Well, so Titty Pat and Brad, you guys are the lifeblood of NeuroMatch. So I want to also put this up here. Let me just do entire screen. I'll share this because this is a list, and now our little windows are covering us, but this is a list of the folks who made the NeuroMatch conference possible. And we're really grateful to all of the volunteers, the showrunners, everybody that you saw on camera and off camera, all the people who did all of the organization, all of the selection of the short talks and so on, all the review of all the talks. I've got more to say, but do you guys want to say anything? Do you have any stats to share?
I mean, I just want to say thank you so much, everyone, for joining NeuroMatch conference from like across the world. I know that we are all in like different time zone, but yeah, I really enjoy the talk, even though it's like in my nighttime time zone, from like 9 p.m. to like almost like early morning. But yeah, it's a great experience. I have been like hosting local meetups, and like me and my student really enjoy a lot by like gathering together and discuss on like flash talk and watching like some talks together. So overall, like really enjoy everything throughout the conference, and I hope like everyone enjoy it too.
Yeah, Titty Pat did a great job of building the interface almost from scratch, basically, with a colleague of his named Tulican. It was really great to be able to have the videos embedded, the agenda browser working so smoothly with the searching. It's just really awesome to have that functionality. And the Discord thing worked really well. Apparently we had over 2,000 messages in Discord and 395 registered users just from the last 2 days. And that's going to keep going. I mean, there's no reason to stop it. So that's really awesome.
Yeah, that's great. So if you haven't joined the Discord server and you haven't been looking at the flash talks, please do that. The NeuroMatch fun continues. There's a button at the bottom of your screen that says asynchronous flash talks in Discord, and it is shiny and green. So you should click that button and go to the abstract browser and click on Discord and join the conversation. Titty Pat, I'm so glad you got to do a meetup. We did a little meetup here at UC Irvine as well. We got together last night for some drinks and snacks and talked about the conference. And then today we had tacos at lunch, and it was great. And we talked about the conference in the flash talk sessions. So I think that's it. I see, Brad, you've got your NeuroMatch shirt.
I've got a question, actually. Is there any reason that we have to take the meetup thing down? I mean, could people still like host meetups and use that as a way to find other people who live locally?
I mean, we could still like have the meetup right there, and then people can like host the meetup, and we can see like a lot of meetups like coming up. I think just have to like specify time if you would like to do the meetup like after the conference to watch flash talk.
But it's just like a really great way for people to find other people who happen to live physically near them. The interface works pretty nicely in that respect.
I think that's a great idea, especially because all the content is all recorded now.
Right
so
there.
Anybody can come back right here.
Time. It's like the conference that never ends.
Literally, just bring up this URL and have a watch party. Just have it streaming in the background of your holiday party, your Christmas party. Is perfect.
Yeah, all your relatives will love it. Christmas, whatever.
Yeah, that's what I do with my student actually. So like I really like some of the talks, right? So I pull it up and then let the student watch these talks.
Our lab is watching a psychonomics talk tomorrow morning. It's great.
We've done that too. Yeah. One thing that I really like about the flash talks too is that you can have two people who meet at a meetup or something like that, and they can both pull up each other's flash talks and walk away from each other for 5 minutes and then come back and have a conversation about both people's talks. It's cool. Like the most efficient way to actually like have a conversation about science. It's great. Yeah. Well, I think that's probably it for us. Brad's got his NeuroMatch Academy shirt. I don't have mine on today, but I do have my Y hat from NeuroMatch. The first days of the. So this was really, really fun. I'm going to take this off because it's so silly. But this was really fun, everybody. Thank you for being here with us. Thank you for staying to the end. Thank you for joining us for computational neuroscience wherever you are in the world, whenever you are in the world, whatever time it is where you are. And enjoy the Discord server, enjoy the conversations, and I'm sure we will see you next time or continuously into the future in the NeuroMatch universe, I suppose.
Yep.
Okay.
All right.
All right, folks. Take care,
everyone.
everybody.
Yeah.
See you next time.